immobilization in industry nowadays. So I'm Nur Afifa.
And I'm Muhammad Nabil.
And I am Anisis Misraya Kudian.
Together today we will discuss about the enzyme application in industry nowadays. Let's know a little bit about enzyme. What is enzyme?
What are enzymes? They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they would be too slow to support life. There are many different types of enzymes, and they come in all manner of shapes and sizes. For example, what you call the piranha enzyme is actually pepsin. It is only found in the stomach and digests proteins. In your mouth, there is amylase, which breaks down starch. In your cells alone are hundreds of enzymes that control your cell activity and defend them from invasions by microbes like bacteria and viruses. Though enzymes can appear very different, they all share a few characteristics in common.
Nowadays we use a lot of enzyme in our industry. So what can you say about the method of enzyme immobilization in our industry?
So basically there are 5 methods for enzyme immobilization, such as adsorption, covalent bond, cross-linking, and so on. So basically there are 5 methods for the enzyme immobilization. The first one is adsorption, the second one is covalent bond, the third one is entrapment, the fourth one is cross-linking, and the last one is encapsulation. So basically for the first one, adsorption, we already always heard the words adsorption. So adsorption means the enzyme will be adsorbed on the external surface of the support within the carrier. There are basically several types of carrier that can be used, such as organic carrier, ion exchange carrier, and also mineral carrier. So in adsorption, there are no permanent bond formed between the carrier and also the enzyme in adsorption. The weak bonds are being used to stabilize the enzyme to the support. There are several advantages and disadvantages by using this method, such as it is easy to carry out and it requires no reagent, while on the other hand the efficiency is less, and also the desorption of enzymes from the carrier. The desorption of enzymes from the carrier, it means that it was not being absorbed on the external surface. So the second one is covalent bond. Covalent bond involve the formation of covalent bonds between chemical groups in enzyme and to the chemical groups to the support or the carrier. This method is widely used compared to adsorption, whereas the adsorption is the most simplest and the oldest method, while this method is widely used. So the hydroxy groups and the amino groups of support or enzyme form the covalent bonds easily. There are several carriers that are commonly used, such as carbohydrates, which is cellulose, synthetic agents, and also protein carriers such as gelatin, also collagen. This method can be useful as it will be no leakage of desorption, and also it is a simple method. However, it might cause to enzyme inactivation. Moving on. The third method is entrapment. As the word is entrapment, means that the enzymes are physically entrapped inside a porous matrix. There are several methods of entrapment. The first one is inclusion in gels, meaning that the enzyme is being trapped in the gel. The second one is inclusion in fibers. The enzyme supported in fiber made of matrix material. And the next one is inclusion in microcapsules, means that the enzyme is being trapped in the microcapsules formed by monomer mixtures. Okay, this method is a fast method and it's cheap, and also it's easy to be conducted at a small scale. However, they might cause a leakage of enzyme. The fourth method is cross-linking. The enzymes are being directly linked by covalent bonds between the various groups of enzyme by using polyfunctional reagents. Okay. In this method, there is no such thing as matrix or support involving, and then this method is cheap, but it's not often used for the pure enzymes, but it is widely used in the industrial. However, the polyfunctional reagents used may be denatured. So yeah, that's the disadvantage of using this method. And the last one is encapsulation, like entrapment. It is being entrapped, and this method, for this method, it was being done by enclosing enzymes in a membrane capsule. The capsule is made of the semi-permeable membrane. This method basically depends on the stability of enzymes inside the capsule. So if you want to know how effective this method is, we need to know the stability of the enzyme itself that we have been enclosed in the capsule. We knew that we use enzymes in the industry. So how does enzyme immobilizations of enzyme works in the industry?
Immobilization technology is widely used in industry nowadays, such as biomedical application, food industry, wastewater treatment, and textile industry.
First of all, biomedical application. Immobilized enzyme widely used in diagnosis and treatment of many diseases. For example, immobilized antibodies, receptors, and enzymes are used in biosensors. This technology also used to overcome inborn metabolic disorder by supplying immobilized enzyme. Effectively used in drugs delivery system to release encapsulated enzyme or protein may be considered. And the second one is food industry. Enzymes like pectinases and cellulose immobilized on suitable carriers are successfully used in the production of jams, jellies, and syrup from fruits and vegetables. Food industry also immobilized enzymes are most robust and more resistant to environmental changes. More importantly, the heterogeneity of immobilized enzyme system allows an easy recovery of both enzyme and products. Multiple use of enzymes and continuous operation of enzymatic process. The third one is wastewater management treatment. In wastewater management, this technology widely used in treatment of sewage and industrial effluents. This technology is used to remove micropollution. Immobilization technology also effective in treating high concentration organic waste water. Many kinds of wastewater contain high concentration and organic matters such as industrial and living wastewater. Last but not least is textile industries. This is including scoring, biopolishing, and desizing of fabrics. A number of enzymes have application in the textile sector, as several natural fibers can be subjected to processing using such natural biocatalysts. As I mentioned before, there are biomedical application, food industry, wastewater management, and textile industry.
What do you think about improvement can be done in future in immobilization technique?
So I think there are many ways or methods that can be taken for the improvement of enzyme immobilization enzyme in future. Although we already have numerous ways of enzyme immobilization techniques, but still we need to have some improvement in getting a robust immobilized enzyme as a catalyst. The new proposed enzyme immobilization technique must meet the catalytic needs, which are working at the desired selectivity, productivity, and also the space-time yield, with the aim of achieving fewer side reactions, high productivity, and also high space-time yield, as well as high durability or stability of the catalyst. Besides, the requirement for the non-catalytic function should not be neglected. Geometric properties, types of reaction medium, reaction system, and process conditions, to name a few. These requirements are needed to achieve easy separation of the immobilized enzymes from the reaction mixture. Other than that, it's broad applicability in different reaction medium as well as to facilitate the process development. So, we already know that the main task of enzyme immobilization technique is to select suitable immobilization techniques which meet both the catalytic needs and also the non-catalytic needs, as I have mentioned earlier. Thus, in future immobilization, it is probably not to develop new method for the immobilization itself, but to know how to achieve the desired performance of an application that we want by selecting an appropriate immobilization technique. For example, is by using the conventional method or the present techniques of immobilization. However, an integration approach or a new design is suggested for a robust immobilized enzyme. So, the future technique of enzyme immobilization is by using nanotechnology or nanostructures, which include nanofibers, carbon nanotubes, and also others nanoparticles. In the beginning stage, enzymes such as trypsin are immobilized on the structure of the nanoparticles, which use a conventional method such as simple adsorption and covalent attachment. This provides a large surface area and thus increase the enzyme's activity. After that, a lot of techniques have been implemented for the increase of enzyme stability, such as nano-entrapment, ship-in-a-bottle approach, and also covalent linkage, to name a few. It is suggested that nanoscale catalysts would be used for the treatment of water contamination in the future.
So today we have learned about what is enzyme and what the method used for enzyme immobilization and the industry that attached with enzyme immobilization technology, and what can be done in the future for improve this enzyme immobilization technique.
I think we have talked a lot today. So, see you guys later.